Okay guys, we're just about to start our next session after lunch. I hope you enjoyed lunch. Sorry the jacket potato was very, very big. So hopefully you're full. We are doing a raffle at the Swallow stand again just to try and raise a few bob. And if you go to the raffle stand in your coffee break, and obviously that'd be great. Just get the raffle tickets. Also on your table now you'll find little yellow slips. Please write your comments and your ideas and thoughts, and we can then put them up on the board. So thank you very much. Just see if are you ready for the two speakers as well after? I'll let them go on the stage.
Right, so how would you somebody like my husband and that's major, that's a condition.
Well, the condition can be being a hospital, it can be being the job.
Good afternoon, everyone. Welcome to the afternoon session. My name is Arthur Loretano. I'm a head and neck cancer surgeon from Boston. It's a great honor to be invited back to speak at the Swallows. This is a tremendous organization, as you've seen, and I can't thank Chris and Sharon enough for asking me to speak. Obviously, they asked me to speak after lunch because people get a little sleepy after lunch and they want me to keep everyone awake. When I was in high school, if we started to get sleepy after lunch, they would make us run a lap outside around the school. I won't make you run around the stadium today. I absolutely promise that. I love coming to the UK. People come up to me and say, "Oh, we love your accent." I know what they're really thinking. They're thinking, listen to the colonists have bastardized our beautiful English language over 400 years. But you're very nice about it, so I'll just leave it at that. I also want to thank Denise, because I thought my ankle swelling was not from traveling, but from all the salt in my fish and chips and all the Guinness I've consumed since I've been here. So now I know it was just from sitting on the airplane. Enough said. So I had slides prepared. In fact, you can read about me. You don't need to hear about me, so we'll leave it at that. And I'll tell you about our speakers in a moment. But I ended up editorializing a little bit, and I want to talk about one thing very quickly before we get into our topic of new drugs and new treatments in head and neck cancer. One word I took from this morning's presentations: advocacy. And if you walk away with nothing else, I think you need to walk away from this conference with that. Patients, you need to advocate for yourselves. You heard that this morning. It's very important you push your healthcare providers. I was speaking with someone earlier today, almost 5 years out from their head and neck cancer, from the end of treatment. They actually had never been told what a squamous cell was, and I actually told them during breakfast. So advocate for yourselves. Push your healthcare providers. Carers, advocate for your partners, your patient. Advocate for yourselves as well, as we heard today. Look for treatment for yourselves and make sure you're supported. Now, why am I here as a healthcare provider? Because we need to advocate for all of you. We need to advocate for you as patients and as carers, for taking care of you. And what we need to do now, which is why these people are up here, is we need to continue to make treatment better. When I started out in residency, all we focused on was cure. Just needed to cure cancer. It's so much more important than that. Of course we need to cure cancer, but we need to maintain and try to maximize quality of life. And we need to make sure that people can function as well as they can. And as we've seen with head and neck cancer, treatments cause ongoing problems, lifelong problems, particularly related to radiation therapy. And I don't want to throw them under the bus, but obviously a lot of the surgeries I do alter things permanently. So we need to try to improve quality of life, looking at patient-reported outcome measures as we talked about today. Even for people who continue to have cancer, who we can't cure, we need to try to maximize some element of disease-free intervals if we can get some remission, or at least to try to keep the disease stable for as long as possible. And then when that's not possible, we need to address palliative care, as we heard earlier from someone here, because we don't give up on our patients when it seems like the treatment's no longer working. We work with them right through to the end, and even beyond that. I still have carers who I stay in touch with, even when the patients have passed away. So I can't emphasize that this is a journey. It's a lifelong journey. Swallowing problems can happen even 10 or 15 years out, where I've had patients have to go on a liquid diet or even get a feeding tube when for the last 10 years they didn't need one because they've had the progressive fibrosis that we just heard about in the previous talk. So This is why it brings me back to say how important this organization is and how much it does for head and neck cancer patients, their carers, and for healthcare providers like myself, and how much I learn and bring back to the United States every time I come here. So that's my editorial. Having said that, we have two wonderful speakers today. We have Dr. Lauren Wood. She is the Chief Medical Officer for PDS Biotechnology. She's going to speak to us about immune therapies, particularly vaccinations against cancer cells. She's an expert in immunology. And then Dr. Sam Lighthouse, who's going to speak, he is the CEO of Lightox Limited. He's going to speak to us about targeted light therapy for topical treatment of cancers. I was speaking with him earlier because I've been doing this long enough that I remember when as a resident we were entertaining the idea of photodynamic therapy and really could not seem to make it work. And when I found out I was actually chairing this panel and saw what he was doing, it just brought back so many great memories, and I'm very excited about what we're going to hear about today. So I'm going to ask Dr. Wood to speak first, and I will take a seat.
Well, again, I want to thank Chris and Sharon for the opportunity to speak to you all this afternoon at this conference. I think one of the things that's really, really critical for those of us who are involved in the enterprise of trying to develop new therapies and interventions is to get patient input early on and identify not only the problems from a scientific standpoint that we're trying to solve, but also solving problems and issues from a patient and caregiver perspective and building that into the process from early on. So again, thank you for having the opportunity to speak. So, okay.
Those are mine.
Those are yours. All right, so then we'll flash through.
And I should say, what I left out there was all the evolution that's happened over time with cancer treatment. Feel free to ask me about robotic surgery, laser therapy, other things that we are doing, but obviously the focus is on these two speakers.
Right, and I'm going to try and leave plenty of time for questions and kind of get through my presentation to be able to have the opportunity for individuals to ask questions. So I'm going to be talking to you all briefly about our study, Versatile-002, which is a study of PDS-0101, which is a therapeutic cancer vaccine that specifically targets the treatment of HPV-positive head and neck cancer, squamous cell carcinoma, and we co-deliver it in combination with pembrolizumab, which is an approved standard of care therapy. I'm in biotechnology, so I have to have a disclaimer slide. These are forward-looking statements. I am an employee of PDS Biotechnology. I always have to do that officially. So what are the challenges with immunotherapy? Immunotherapy has really become the fourth cornerstone of cancer treatment. It's phenomenal. We now have surgery, we have radiation, we have chemotherapy, but immunotherapy in the last 10 years has really revolutionized the treatment of cancer, and it's become the fourth cornerstone. In addition to checkpoint inhibitors, which are a major component of immunotherapy and adoptive cellular therapies like CAR T cells, one of the problems is that they're limited in their ability to actually help patients' immune systems promote a strong killer T cell response that will go and attack the tumor. So as a consequence, what happens is that not enough patients respond to treatment. Only about 20%, 1 in 5. So if you are a responder to immunotherapy, it's incredible. The problem is that not enough patients respond. So we've got to move the needle so that a greater percentage of patients are able to respond. Many of the approved interventions often don't delay disease progression, okay, even if they improve survival. So you can have improved survival, but if your disease also evolves with it, that really impairs and impacts your quality of life in terms of you're living longer, but you're living with additional disease symptoms that you didn't have previously. Oftentimes, the approach always is in cancer, if you have activity with one agent, let's give more, let's give lots more, and sometimes you've got lots of agents that have lots of side effects, and when you look at the relative ratio of how much benefit you're getting from the combination versus the adverse effect profile, it's very limited. And those impact quality of life. The thing that I just want to highlight about the standard of care is pembrolizumab, which is also known as Keytruda, made by Merck, has really been shown to be differentiating in the treatment of patients who have first-line recurrent metastatic head and neck squamous cell carcinoma. And that's why we're delivering it in combination with PDS0101, which is our therapeutic vaccine. So as the CMO, one of my charges is in the clinical development of any combination therapies, it's like, how should we be trying to address the current unmet medical needs of cancer patients and their caregivers? Well, we want to help more patients benefit from the therapy, so we want to move the needle from 20% to 35% or 40%. If we go from 20% to 35%, then that would be 1 in 3 patients benefiting from therapy as opposed to 1 in 5 patients benefiting therapy. And again, we want to get to the point where we can help the majority of patients benefit from therapy. Delay disease progression, because that affects quality of life very significantly, and we know that there are a lot of residual treatment effects from standard of care treatment. Delay the need for chemo, if at all possible. That's one of our goals, because we know that the effects of chemo can be long-lasting, prolong life, and importantly, prolong life while improving quality of life. So I'm going to just highlight for you what Versamune is. Don't be overwhelmed by this slide. I'm an immunologist. I get very passionate when I get a chance to talk about immunology, but we're just going to go from the left-hand side of the slide to the middle part of the slide to the end. And there's a quick takeaway message in each component. Versamune is lipid nanoparticles. Maybe a lot of people have heard about that in the press because all of the COVID vaccines that have been licensed globally that have made a difference, they are mRNA, but they're coated in lipid nanoparticles. And lipid nanoparticles just help things get into cells. And so this lipid nanoparticle, called Versamune, is sized to be about the size of a virus. And then we can deliver different pieces of tumor proteins with these lipid nanoparticles to help the immune system get activated. So that's what's on the left-hand side of the slide. You just have the nanoparticles plus the immune system proteins, and in the case of HPV-related cancers, it's pieces of the HPV, human papillomavirus, and it causes cells of the immune system to take up those particles and process them. Well, in the middle side of the slide, the cells that take up the antigens and process them, they then present them to cells of the immune system. They're called T cells, all right? You got T cells, you got B cells, we got so many complexities to the immune system, but the key thing is that it trains these T cells to recognize the enemy. And what is the enemy? The enemy is those tumor antigens that are on that squamous cell carcinoma that for some reason the body is not rejecting. So the body has to be trained to say, hey, what does the enemy look like? Train it, and then go and attack it. So that's what's happening in the middle, is that we have helper T cells, we have killer T cells, and the bottom line is that they're trained to then recognize a patient's specific tumor. But it's not only about having the cells, because, you know, cells that sit on the sideline aren't going to get the job done. You got to get across the finish line. You got to get to the goal. So those cells actually then go to the tumor and are able to destroy the tumor. And that's the whole principle behind this therapeutic vaccine platform, is to stimulate those T cells, make lots of T cells in the body, then able to go and recognize the tumor. So what's PDS0101? It's a therapeutic vaccine, okay, that stimulates T cells, what I just showed you in the previous slide. It's given by a simple subcutaneous injection. One of the things that we have in our study is, because I deliver a vaccine, we really have to capture the side effects. It's like we really want to know how well you tolerate the vaccine, what are your local injection site reactions. Well, for many people who have undergone very intensive resections, they've undergone chemo and radiation, they're kind of like, this is a breeze compared to chemo and radiation. And we have a hard time. Just remember to tell us how long you had an injection site reaction and how long it lasted. We have some interim data that I'm just going to highlight for you that suggests that when you deliver this vaccine in individuals who have HPV-related cancer, that we can stimulate the immune system to actually do exactly what I highlighted, and that is to train T cells to attack the tumor, to shrink the tumor, potentially delay disease progression and prolong survival. And importantly, when we give this with Keytruda, which is the standard of care, we really see no evidence that there are more toxicities or new toxicities associated with the combination. So that's why we're trying to explore things and keep things moving. So I want to say welcome to our versatile, whoopsie, is that me? Our UK and Ireland Versatile 002 sites. We recently expanded this clinical study to here in the UK. I want to say welcome to Dr. Srinivasan, Dr. Metcalf, Dr. Harrington, who will be speaking tomorrow afternoon, as well as Dr. Grant. And we're really, really excited to be able to make the study available here in the UK to patients. Am I knocking something? Sorry about that. Apologies. Maybe I'll stand away farther from the mic. This is a summary of the study. The key takeaway is this. We're studying the combination in patients who have never seen pembrolizumab. So they have recurrence of their head and neck cancer. It's either unresectable, recurrent, or metastatic, and they've never received pembrolizumab. They are able to receive the combination therapy, and we're investigating in those patients whom we call checkpoint naive patients. And then we're also investigating the combination in patients who have seen pembrolizumab alone or in combination with other therapies or even other checkpoint inhibitors that have been approved, and they've progressed in their disease, because the thought is maybe the reason that they didn't respond initially is that they didn't have enough T-cell gas in the tank. And we want to see if we can deliver a therapeutic vaccine that augments the T cells. This is a scientific slide. The key takeaway here is what we are obligated to do when we do clinical trials and we are measuring and trying to measure the effectiveness of a therapy. We basically want to see if we can shrink the tumors. So everything to the right of the slide is good, because that means there's been some level of tumor shrinkage, and that's what we always want to achieve. This is data that we presented earlier in the year at a big global oncology meeting that happens every June called ASCO. But on the right-hand side, you can see green bars are complete responses, blue bars are partial tumor shrinkage, gray is some tumor shrinkage, and then we've got individuals who progress, who don't seem to respond at all, and those are red on the left-hand side. The key takeaway here is that preliminarily, we're seeing that approximately, well, I don't know what to do. Preliminarily, we're seeing that approximately 41% of patients are able to achieve a response. And so this is very early data, it's preliminary data, but remember that first bullet I talked about? Let's see if we can move the needle and help more people respond to therapy. That's one of our goals. The other thing is that if we can't shrink the tumor, that's always the first objective, but if we can stabilize the tumor so it doesn't grow as quickly, then we can delay disease progression and then hopefully also improve quality of life. So on the left-hand side, big complex table, the takeaway message is that we have local injection site reactions, which you would expect to see. You give a vaccine, people are going to say, oh, got a little bit of discomfort, I've got some swelling, I've got some redness because I've got an injection. But the important thing is that we haven't seen any unusual adverse events associated with our vaccine in terms of severe allergic reactions, and certainly there's been no new change in terms of the known profile of pembrolizumab. So our goal again is, if we're going to give combination therapies, let's give combination therapies that can work together, but also have not only better anti-tumor activity, but at least let them be well tolerated so that we don't get more side effects associated with things. And then another key goal is to delay disease progression and prolong survival. And in this preliminary group of patients with recurrent disease, almost 90% of them were alive 9 months into receiving treatment. Over half of them had not had any evidence of progression, and the overall survival rate was also very high. So again, we want to see if we can continue to help these trends be confirmed. So that's it. I think I took less than 15 minutes to be able to have Sam give an opportunity to speak. But those are the principles behind this trial. And again, I really thank Chris and Sharon for the opportunity to invite us here, because there's been revelation after revelation with each set of speakers in terms of things that we need to do as far as getting input from patients and their families, as far as what's feasible, what's practical, what kind of problems need to be solved for you guys who are on the front lines. So, thank you.
So hopefully my slides are up next. I'll repeat to Chris and Sharon for putting this on. This is probably the most nervous I've been in the last 4 years. You can put me in front of investors, you can put me in front of a scientific audience, and I'm fairly comfortable there. When it comes to patients, it's a lot more emotional. And I'm a technologist, really. So I'm a chemist by training. There is no chemistry in this, you don't have to worry. Molecular biology PhD, there's people even from Sheffield. So, but I moved out of that. My friend died 27 years old. He had an aortic aneurysm. And I moved into medical technologies because I wanted to see how I could help and what I could do. And I started putting stents into people, which are little scaffolds in the heart, and then putting drugs into those stents and letting them dissolve so that the injury site was less. Then we moved into putting stem cells onto them because that was cool at the time. And really from there it develops out. And I was okay at building businesses from sort of scientific research into small teams, into larger teams. HPV is a very interesting area. My last company, we developed point-of-care diagnostics. So if you have let's say, I don't know, a respiratory disease like COVID, you can take that sample, 20 minutes later the box will tell you through PCR, because everyone knows what PCR is now, whether you had COVID or not. But we developed that. We had a lab out in South Africa that was looking at HIV and TB. And then we got in touch with the Gates Foundation and they said HPV would be much more interesting and you could do that in India. So we built the company and put it that way, and 110 people and 100 million quid. And then COVID happens and you pivot very quickly because there's a lot of money to be made over there, but the HPV stuff's still there. And it's really interesting because HPV keeps coming back, whether it's in cervical or oral cancers. Links between that and disease and everything else are really, really interesting. So one thing I found with head and neck, and looking at this from a point of view of, if you go back to when I was putting things inside people's hearts, we did it through a tiny little cut in your femoral artery, and then we put a catheter all the way up and into the last bit of your heart here. And then you inflate a little balloon, and you whisk it all back out again, and that was it. You know, the patient would be in and out within the same day. And you think, if we can develop that type of technology, surely we'll be able to do interesting stuff in other places. And I heard earlier, head and neck, it seems to be forgotten or not as in the press, or I don't know, it doesn't have the right attention that it should. And considering the stories we've heard so far, and it's devastating, it's really difficult when you're developing something not to really want to push it as hard and fast as you can. Developing drugs is all about safety. Can't put anything into you unless it's safe. So then we have to prove safety, and then we can start to expand where these things go. And I think that's the thing that you really want to do. So I wasn't very well. I had gastrointestinal surgery. My mate, who's a chemist, was developing these molecules, and Carrie, who's at the back of the room, was working with him. She's a professor in Durham. In fact, they're both professors in Durham. Much smarter than me. And while I was recovering, so I had 2 months off work, and thought I was a captured audience. So he sent me a business plan for Litox. Now, everything that you've seen in this area so far has either involved cutting something open or shining some sort of electromagnetic radiation on it, or adding a chemical to it. Okay, and Litox is basically a combination of all of these things. Light-activated technology, so how does the chemical, the light, and the biological person interact with each other? And if you keep that in mind, that's really important to what we're going to go through, because essentially what we're looking at is by accident, and science is best when it happens by accident, we were looking at fluorescent molecules, same sort of things that Illumina used to sequence, where they go in a cell, why they go into certain places in the cell, what they do when they're there, and we have the chemist in Andy, and then we have Carrie, the biologist, and Carrie's a wound specialist, and cell plates, and the technician looks down and all of the cells are dead. And she thinks she's messed it up, she throws it away. Does the same thing the next day, comes in, gets the cells ready, puts the drug on it, what we now call a drug, then we call it a chemical, goes and makes a cup of tea, because technicians are creatures of habit, comes back again, dead again. What's happened here? Well, the drugs that we made are photosensitizers. So the cell isn't happy because it's got the drug in there, and the microscope light has basically activated the drug and burnt a perfect hole where the light was, but all of the surrounding cells were still perfectly intact and perfectly happy. So the drug is basically only active when it's in a cell. It won't work outside the cell. When you have the right light on it, because it has to be a certain wavelength of light, and then it's in the right concentration. So then you take that forwards. Now I don't need to tell everyone in here, so I'll skip over this because you guys know how this works, and it's kind of devastating too. But the things that I think are important, things like, you know, when you use radiotherapies, it's not very targeted. Okay, when you're using chemotherapies, you're putting all of the drug into all of the body and all of the side effects that go with it, and the surgery, in the nicest possible way, is pretty brutal and pretty primitive. And we looked at this and said, well, okay, how far can we take this, and where are we looking for? And we look at all the things through why we would want to treat these areas, and what do people see? How do you present? And you get these types of images, and you know better than I how this works. And we look at that, we look at our drug, and we say, well, how do we get our drug to this? And luckily, our drug is small. So literally, we can put it on in the right area. It's not happy. There we go. So when we look at this, and then we look at why we want to do this, and you can think of all the application areas where if you can get a drug and you can get a light to it, you might be able to help someone. Carrie's a skin specialist, a wound specialist, so skin is very obvious. We had a chat with a friend of mine in Sheffield who's head of clinical dentistry there, and he said, well, I'll take it, I'll have a look at this. And this is the sort of pathway you get. And for some people in the audience will have been here, but this surveillance bit I thought was really, really interesting. You've got one of these things and we don't know what to do with it. So we'll just leave it and wait. Or we don't, and we just cut it out and everything that we've heard, and the devastating side of that. And there's no other part to this. So that's what we want to be. We want to be the other part. So I've told you how we make drugs and how you develop those, but essentially the way that we've developed the drug is to apply it to the surface. It targets certain cancerous cells. They have things called HDACs, which I don't need to go into, but basically cancer cells have great ways of sort of showing you that they're cancerous from a molecular level. So we can target those in, and then when you shine a light on it, the drug becomes toxic, as I said. But that toxicity is really important, because it's essentially looking at those cells and saying, these shouldn't be here. And the way the drug works is it's a natural cell response. So the light is telling the cell, or the light is creating a chemical within inside the cell, it's called a reactive oxygen species. Everyone knows about antioxidants. Well, we want to do the other way. We want to really ramp up those oxidants in that cell. Then the body will say, that cell's not going to survive, it will die. Cancerous cell, it will die. And then your body hopefully will go, that shouldn't have been there, let's regenerate good, healthy tissue. And we don't know that we'll do that because we have to do that. But obviously this then gives very clear benefits to what you will be able to do. So hopefully this works and we reduce the amount of surgery needed, or hopefully avoid surgery altogether. And it's a first-line treatment. So hopefully these cancers don't progress further and that we're treating them earlier. In the same way that you want to diagnose early, you want to treat early, you get better outcomes if you do that. And no one wants to wait. So all of those things. And I think the one thing we learned from all of the conversations we've had is the quality of life in this area in particular is so drastically reduced when you go through these surgeries. But that's not always recorded in data, and I think that's absolutely shocking. We're not the only people that do this stuff. You know, when you mentioned phototherapies earlier, you know, my PhD, whenever that was 20 years ago, was in porphyrins. And everyone else on this slide, other than us, are using porphyrins. And they're basically your molecule that is your hemoglobin in your body, and you're creating these things. But the problem is you photosensitize the whole patient. So they're looking at different areas, and there are lots of links into this that are interesting. So Rakuten, the logo you might notice, is a Japanese billionaire, so he's put 300 million into the company. And they're looking at the lower oral tract. Aura have actually taken an HPV vector and target into the ocular cancer area. Steve is looking at urethral cancers, Photocure as a diagnostic device. No one is looking at where I think is the most obvious area to start, because all I have to do is get the drug to the area and use what is essentially a dentist curing light to activate the drug. Why that is, I don't know, but hopefully this will all come through. So we want it to be really simple. These are some faces of very clever people and me. So Carrie I've mentioned, Andy I've mentioned, Mark is an immunology professor, because if you're stimulating an immune response, that's really important. Roy's made drugs all of his life. Paul and Franz sell drugs for a living. These people are much more interesting. So we were chatting with Mike Nugent up here, and he put us in contact with people in Liverpool. And these are the people that you actually really want to know. Because it's all very well me saying I can develop a drug and I can give you a light and blah, blah, blah. But if these guys can't use it, it's absolutely useless to anyone in the room. Because you have to develop the drug so that people can use it. You don't want it to be a mouthwash, because you don't want to, you don't want to photosensitize the whole mouth. You want to be able to literally put it on. So I use it as an analogy. Imagine a fluorescent Bonjela gel, that when you put that light on it, it stimulates it in the area that you want. So lots of control, and hopefully minimal damage to any tissue outside. Now often people present with multiple lesions. Well, that's fine, we'll just treat multiple lesions at the same time and hopefully get regression there. And that's why these guys are so important. So hopefully, and the company's at pre-clinical stage, so now we're doing this in animals and in cell models and things like that. But these guys will get hold of it next year, hopefully, and actually start to put it into people. And yeah, that's really the critical part. So this is what we do. Again, it goes back to, developing drugs is a long process. And if I mentioned stem cells at the beginning, and now you see stem cell therapies, that's about a 15-year journey. These light-activated drugs have been around for 30 years, and the old style of them, and hopefully we're able to then bring these forward and bring them into new ways of treating people. And then ultimately, if we can do this in the head and neck cancer space, which is where we are at the moment, then we're able to then take that data and then look at other areas as well, whether that's skin or cervical or bladder. And then you can expand your portfolio. It's called expanded portfolios, but essentially you're able to help a lot more people. And I think that's something that we don't do enough of, in terms of we have a lot of drugs that aren't repositioned, because we're looking at a drug in isolation in a clinical area rather than from a biological point of view that can actually help. But that's a regulatory grumble. So I will leave that there. Hopefully that's interesting. And thank you very much for listening.
We're going to open up for some questions. I actually have one here from the Twitterverse. Not from Elon Musk. Not sure. He might have fired himself for all I know. I think you already answered these, but this is for Dr. Wood. If a patient has been treated with Keytruda for 2 years, can they now be treated with Keytruda and Versamune?
And the issue is yes, but what we want is individuals who have progressed on Keytruda. So if they stopped their 2 years worth of treatment, they did well, or if they had to discontinue treatment because it looked like their disease was coming back, then they fall into that group called refractory checkpoint inhibitors. So yes, they would be eligible.
All right, and then this is all from the same person, which you may have answered already. Does a combination of Keytruda and Versamune offer greater efficacy than Keytruda alone?
So we are looking to see if we have the start of a signal that maybe they might work together better. We ultimately prove that when we go to the next stage and we compare delivering PDS0101 and Keytruda together versus Keytruda alone. But right now we're in that early stage of delivering them together.
And then the final question you answered with your map, which is will the Versamune trial be expanded to UK participants?
And the answer to that is yes. And I did not plant that because I don't even have a Twitter account.
So, you know, I mean, this is about new treatments, new drugs, and obviously we want to focus on these. There's a lot happening on the diagnosis side, on the treatment side, and then we've already heard a lot about the post-treatment side in terms of lymphedema treatments, things like that. There is now a blood test that's begun to be used in the United States for HPV-positive cancer. It's something we're just starting to entertain using in our own place, and there's pros and cons. It's not a screening test, you know, it's not something to see if the average person has it. You can go to Amazon and you can actually get mucosal testing to see if you have HPV, not HPV cancer, but just HPV exposure. So these are things that, you know, are helping in the pre-treatment phase, or in the diagnosis phase, I should say. So in terms of the treatment that we've heard about today, patient comes into my office and says, I heard about this light therapy, when can I get it? These are questions we get, and then it's the same thing. I've heard about this vaccination. When can I get it? I read about it on Google, and we get this all the time. So, Dr. Whitehouse, what can I tell my patient?
Not yet.
Which is usually my answer.
That's a very simple one, isn't it? No, I mean, so the drug journey is always long. So these molecules and the story I tell about the cells dying, you know, that's nearly 8 years old. I've been involved in the company for 4 years, and we've been doing a lot of that safety testing, and you can't put it into a patient until the regulatory bodies say yes. We're talking with them at the moment. Then you have to, as we've seen before with medical cannabis, you have to make it to the right standards. Now we can do that. We can do that actually about 30 miles down the road from here, and then we can start putting it into patients. Now the problem becomes the amount of money that you need to get through all of the clinical trials. So with a good wind, you know, if I was Elon Musk, then you can push it really quite quickly. I think one of the things that the pandemic has done for us is really kick the regulators up the arse a little bit. You know, there are a lot more things we could do if the regulators let us. Now, this isn't, you don't want a Wild West scenario, absolutely not. But you have to make sure that actually these things are progressive. And you see, I think another thing that the pandemic has done is highlight the way in which the industry now works. So a lot of people still have this idea that large drug companies are the only people that make drugs. Simply not true. What often they do now is they get involved once a smaller company has shown viability and safety and efficacy. So they would rather spend £500 million buying a drug once it's been through 30, 40 patients than all of the research that goes before it. So that's the sort of hurdles that we have to get over. So the answer is still not yet.
That's fine. It's better than my saying, I have no idea what you're talking about. It's usually pretty embarrassing. They show me the website on Google and say, you should know about this.
Dr. Wood, yeah.
So we're farther along in the same thing. I can highlight the journey in terms of at least 10 years in terms of really trying to understand how our specific platform worked. Because part of putting in that time and understanding about your technology is that the better that you understand it, then you try and have an idea of what may be its limitations when we actually start to test it in humans, what we need to watch out for, and how we can optimize the therapy. So we have an understanding of the fact that we didn't really see any toxicity with our Versamune platform. We understand how we have to deliver it with certain proteins that come from the tumors. I love the idea of the phototherapy because, again, what does it do? It specifically attacks where the problem is and leaves healthy, normal tissue. Because so much of what our treatment of any kind of cancer is dealing with, it's not only about killing the cells, but then it's all about the additional residual side effects that we get on the healthy, normal tissues that we have to address in terms of being at risk for infection, diffuse inflammation so that you can't swallow and you get mucositis and so forth. So being able to develop therapies that are very, very specific is really important. And we're at that stage, you know. It's safe. So we've had that first hurdle. Is it safe to give with something else? Looking like we're getting to that hurdle. Is it more effective than just both things alone? Looking like there's that signal. And again, once we get to a level where we have confirmation that things are trending the same way, we go to the next level and say, head to head.
Yeah.
Which one wins?
And I think it's really interesting. So a lot of the time drugs are developed without actually understanding where they're going to go and what that pathway is going to look like. And the slide where I showed the clinicians is really interesting because what you do is you then understand what the patient has to go through, and you know, some of the stories about how many journeys you have to go backwards and forwards to hospital, and I mean it changes your life very, very quickly. And our Caroline, who's up there, said, you know, this isn't really a treatment; it's almost a prevention. But I can do it where the patient, you know, is literally sat in a chair, so it feels much more like a dentist than it would an oncologist trip. And I think that's really where you have to. I think now we have to start demanding more from these types of drug development pathways and involving the patients inside that development as to, you know, what is this actually going to mean? How devastating is it going to be? And I think particularly in this, it's such a devastating change to your life. You know, how do you make sure that this monitoring process is manageable for everyone? You know, once you start developing these lesions, more than likely you continue to do that for a long time, if not the rest of your life. So that's a huge commitment of time and money from both sides, and I think that's something that hopefully we can start to change if we do bring these things through.
Sounds good. And just before we take questions, one of the things that's sort of been a backdrop to what we've been talking about today is how much do we damage normal tissues? I don't know if you saw it on one of the slides, but there was mention of when we do surgery, we have to get a margin of normal tissue, 1 centimeter to 2 centimeters. It's a little bit of throwing the baby out with the bathwater. We have to take some normal tissue out to make sure we've gotten around the abnormal. When we do radiation, we're damaging normal tissue while we're in the process of killing the cancer, which is why you can't re-irradiate in most cases a site that's already been radiated, because then there's too much damage. So what we're hearing here is a way to try to really focus robotic surgery, same idea. Try to remove as little normal tissue as possible and maintain function, and that's what we're hearing in these type of treatments. So let's open it up to questions.
Yeah,
Other questions?
well, you know, what I'd like to say is that with patient advocacy, And I know NICE is the ultimate way you're going to end up in front of a NICE panel sometime in the future. I went on a training course with NICE and they talk about the light bulb moment. And what I think drug companies and your inventions or any inventions, at that light bulb moment, there should be an advocacy on the board. Because if there isn't an advocacy on the board, you can spend a lot of money. And I'll give you an example. I went as a patient advocate against another drug company that was trying to bring a drug in for head and neck, which was fantastic. And it had some great results. But then I said, well, most patients struggle to swallow. And I said, so what does that make a difference? Well, you're asking them to swallow 6 pills. What do they do if they don't swallow? And they said, well, they won't be able to continue. But surely every patient can swallow. They hadn't thought to ask the patient whether you could swallow or not swallow. So NICE then said, would you like to take it out of the room and decide what you want to do? They decided not to progress, and they went back to the drawing board, and now it's available, same drug, but through a needle. How much did that cost them in money, whereas if they'd have had an advocacy from day one, that problem wouldn't have occurred. And also, in front of NICE, suddenly you're made to look stupid because you really don't know the patient where that drug's going.
Right.
And I know with PDS you've got involved with me right from day one because I've challenged you all the way along. And I think that's where advocacy sit with people like yourselves. And if you don't, I think you are really missing the trick. I really do. And I think, you know, when I saw your board up there, I was frantically looking around where the patient advocacy was and I didn't see one.
Yeah.
That then worries me.
Yeah.
Because you're going to go all the way down that long line and suddenly someone's going to say, well actually, why didn't you do this?
Yeah. And I think it's really interesting. So myself and Carrie, so the Dutch investors are actually a group around that. Myself and Carrie went over and we sat in their house raising money, a small amount of money to do some formulation work, which is not cheap. And we sat there and we were talking about why oral cancers, why we're looking at that and everything else. 2 hours we spent doing this. And the guy sat opposite us, literally turned around and went, you've missed so much of this. And his wife lost her tongue at 36 years old to the cancer. And it was really humbling, first of all, but quite interesting to spend the next 2.5 hours learning all of the things that changed in his life and her life after that. And I think it's incredibly important. And Chris, you're more than welcome to come to our board meeting.
Yeah. So any questions from the floor? Yes. We've done a film once together, didn't we? We won't tell what film it was, but we did a film once. And he was a great star in a film when he was at his university, so nice to see you here again.
So, regarding the cancer vaccine, do you have any stumbling blocks when it comes to the heterogeneity of tumours? And how do you overcome that? And, well, if there is a, have you considered bivalent cancer vaccines? And
So that's a great question. So one of the things that we know about tumors is that they're very, very dynamic, okay? There's just not one feature and one element, because we heard earlier this morning the importance of being able to look at the DNA, the genomic sequencing. But again, the picture that you see depends on how you look for the picture in terms of which portion of the keyhole you're looking for, how big it is. And so right now, PDS-0101 targets the most common HPV that's associated with cancers, okay? A high-risk HPV called HPV-16. But there are over 100 HPV types, okay? And there are 14 types that actually are associated with cancers. But 16 is at the top of the list. And then after that is 18. So that's why we're targeting 16 first. And that's the need to be able to deliver a vaccine that will kind of cast the broadest net. importantly, because we're targeting a specific protein that's associated with HPV, and HPV can cause not only head and neck cancer, but it's also associated with cervical cancer, anal cancer, vulvar, vaginal, penile cancer. So this is a way where we're kind of going after a molecular target. But the important thing is that we know that one size never fits all. And that's why there's a goal to have a second generation vaccine, hopefully down the line, where we would include not only 16, but 18. The other important thing that you kind of highlight is, well, how can we maximize the activity of any agent? And this is why I love immunology, because the immune system is infinite. So it turns out that if you only give small pieces of protein, then you kind of narrow down what the immune system is able to respond to. Okay, I know, it's me and my mic, I can't help it. But if you give larger pieces of protein or whole proteins, or there have been some studies of cancer vaccines that actually include killing tumor cells and so forth, what you're able to do is the immune system itself then has to try and respond to many, many, many different pieces of, okay, thank you, Chris. The immune system itself has to respond to many, many different pieces of the tumor. And that way you deal with the fact that there's different aspects. So I'll use my fist as the tumor. It's a mass. We want to get rid of it, right? Well, if I only give you the little piece of the protein that is this knuckle, then that's all your immune system is able to recognize. But if I kind of give you pieces and chunks of this tumor, then your immune system says, this is what the enemy looks like, and it not only attacks one point, but it attacks multiple points. And that is the goal of trying to harness the immune system with cancer vaccines. And that is that you basically want to bully up on the tumor, you know? I don't care which cells do it. I just want to make sure that there's lots of them, that they recognize different pieces of the tumor, and that they're qualified and capable to get the job done and kick the tumor's butt. So that's a great question.
Another question? Any questions? Oh yeah, come on.
Yep.
You'll be the good looking one.
Question for Dr. the White House. Thanks for the interesting presentation. And Caroline actually worked as a junior when I was training as a registrar, so say hello to her as well. What results have you had so far when you've tried this in terms of resolution of dysplastic lesions? And has there been a difference in sort of different grades of dysplasia? They sort of responded differently to the treatment?
So at the moment, so we've been here's an interesting part. So we start with cells and we work our way up. So we go through cells into animal tissues. There are one of the things that I think is really interesting. So in order for you to put it into a human, you have to show a model, an animal model, that is good and representative before you put it into a human. There are very few good oral cancer models. So we almost got to the point where we thought we're going to have to build an oral cancer model before we even are able to then show that this works properly. So you work around this, and there are models. There's one called 4-NQO, which is basically something you put into the drinking water for a rat, and they develop these oral lesions over about 8 to 12 weeks. And then you can treat that. So you have to almost show the model working before you can then show the drug working, which is why this takes so long. The other way of doing this is actually some very generous patients then donate that through as well. But generally, in our case, we're looking at a fairly early stage, and the tumours that people are donating are generally, stage 2, stage 3 cancers after that. So there is a challenge, and I'm sure you're aware of that. But I mean, Carrie will tell you in great detail about those challenges and how we develop those models. The thing that I think is really important behind that, though, is as a commercial company, you shouldn't try and profit off models. I think that's, people do, don't get me wrong, and it is a perfectly viable business model. But I think if there isn't one in your field and you develop one, make it available. It's not core to what we do. So if we do find something and we find something better, then that's the best thing you can do, because then other people can work in the area. Create platforms and let other people build off platforms.
So, one of the other speakers that was going to be here, and I'm going to question both of you on it, unfortunately he got COVID and couldn't turn up, but they're doing, instead of using mice, they're using flies. So the idea is, and I went to their laboratory and it's unbelievable, these are little flies, and I mean you can hardly see them, and what they do is they separate the male to the female, and they make males become females, which I think is so unfair. But that's the way they've done it. But with these female flies then, they can implant a very minute part of a tumour into each fly. The flies are then mixed, and then the drug is put into the flies, and if half the flies die, then the drug's not working. But if they all die, then the drug will work on that tumour. And they've tested it and worked it, and it's incredible. And they've already saved 6 people's lives that got told they couldn't find a drug to deal with it. And one of the drugs ended up being a drug for pancreatic cancer, but it killed the tumour that was in his throat, and he got told he got 3 months to live. It's incredible. So maybe when you're looking at these things, rather than thinking about mice, maybe you look outside the market. And this guy, this company is in London, and they're using flies. So again, that might be an opportunity. It's a shame he didn't come here because it's phenomenal. So I think there's other ways than just thinking the standard, let's use the mice. And what I did get told when I went to a research conference, in head and neck they only use black mice, because black mice are the best for head and neck. It's the way they use the mice. They don't use white mice, they use black mice. So I just think it's incredible when you start looking at the challenges you have to go through to give me a new drug or tomorrow's patient a new drug. But yeah.
The one comment that we all say all the time is that, I mean, how many millions of hundreds of millions of mice we've cured of cancer. But it has not necessarily translated to a clinical benefit in humans. And so that's why we really need, like a major paradigm shift to go, wait, there is a different way that we can look at the activity and the potential activity of a new agent or a new drug, and rapidly, quickly. I mean, flies multiply like crazy, so you get your answer quick.
Well, that's the idea. What they said is obviously with mice, you're going to need a bigger piece of tumour to get across all the mice. With flies, they are only fruit flies, so they are very small. And literally, I watched them put this new drug into a whole circle of these flies, and all those flies survived. That drug was then going to be taken over to a patient in America to be used against his tumour, because it proved, guaranteed, that it would cure that tumour. So I think flies have got to be looked at.
Anything that reduces the need for that. I mean, animal models are incredibly expensive, and they take an awfully long time. The stem cell work that I did in Sheffield was trials on pig models. And it gets very, very expensive, and often this is a case of what regulations we need in order for it to be satisfied. And then also there's a lot of repetition in the regulating world as well. So, you can do everything that you want for the regulators in the UK, to the MHRA, and then you can go and speak to the FDA and require something else. And then you're adding in more cost and more delays and everything else that goes through it. The difference, I think, really has to be in order to make this a more cost-effective route is to actually get regulators to start working together. Now there are certain areas where they're beginning to do this. So Project Orbis, the FDA launched, and that's chemotherapy drugs, where if you submit through the FDA, you get the UK, Brazil, Canada, Australia, Singapore, and a few others. And that's where the regulators are beginning to work together in order to do this. And the same is true in diagnostics and standards there. If we standardize, and it's just, no one wants to pay Apple for another lightning cable. If we start standardizing the processes by which we can submit new drug filings, then it will become a lot quicker to be able, and a lot more cost effective to bring new treatments through.
So what I really like about this is you're talking about light. You know, Thor is talking about light, but a different spectrum. On the innovation table, I don't know whether you've seen it, there's a little torch. That is, again, if you speak to Dr. Hess and Michael's there, it's to show lesions up with a torch before the naked eye can see it. So light has got a big play in our next journey. So it's great to hear you, and if you want to have a look at the torch that's on the innovation table, let me know and I'll show it you. Because that is the next way that dentists are going to find lesions in the mouth that the naked eye can't see. And the earlier we get dissected, the earlier we're in treatment, the more they'll survive. And I think it's the way forward, and I love what you said. Sorry I took enough time.
Just one thing, if you want to expand light in terms of laser therapy, one of my mentors, Steve Zeitels, who has operated on Adele, and Steve Tyler from Aerosmith, is focusing right now on lasering the blood supply to laryngeal tumors, early laryngeal cancer, so that the cancer just dies and leaves the voice box otherwise completely normal. So there, you know, what comes out of this lecture, or this seminar right here, is hope. There are just so many different things that we continue to do to make patients' lives better. With that, I'll thank you all for your attention, and enjoy the rest of the afternoon, the rest of the conference.
Give me the mic. Can you? Have we got the mic? Yeah, thank you. Right, okay, so a couple of things. You might have seen me going around at lunchtime with yellow post-it stickies on your table. The idea of this is to make sure that those post-its contain your ideas, thoughts, suggestions, comments, and they get put on the feedback wall. All you have to do is have a think about what your comments are, your ideas, thoughts, suggestions. If you want to leave them on if you want to put them on the wall, that's fine. If you want to leave them on the table, that's fine. But can you just have 2 minutes now before you go to coffee? Just have a little thought about that. I know some people have had questions, they've not had a chance to ask them as well, so it might be a question that you've not had a chance as well. So if you could do that just over the next couple of minutes, and then you're free to go for coffee for half an hour. The next sessions, before people go to breaks this morning.
Hi everyone, we're going to start the next session now. If you want to take your seats, that would be fab. And we'll get the speakers on stage and we'll start again. 2 more good sessions to come now, guys, so, and then I'll let you go home. Or you can go to the speakers' dinner, the bar's open.
Hi everyone again. As you can see, I'm not Paul Schneider. I do have many magical powers, but changing shape or form isn't one of them so far. But Paul is not well, so I'm covering this session for him. But also quite pleased because it's an area, this latest research in head and neck cancer, which is quite close to my heart. I dabble into research myself, so I'm quite looking forward to hearing from both of our speakers. So the first speaker is Dr. Jennifer Dean, who's a researcher at the Newcastle University, and she's interested in the early diagnosis of cancer, in particular head and neck cancer. And she's currently working at a project looking at health literacy and how that could impact the early diagnosis of head and neck cancer. And our second speaker is Dr. Kirsty Taylor, who's a head and neck medical oncologist at the Northern Ireland Cancer Centre in Belfast. And she's aiming to characterize the longitudinal changes in the DNA of patients receiving treatment for recurrent and metastatic head and neck cancers. So thanks a lot both for giving up your time to come and join us here. And without further ado, I'll pass it on to you.
Hello everyone, it's lovely to be here today. As you'll see, I'm not the person in the programme, but hopefully you're not too disappointed. I'm just going to aim for less disappointment, if that's okay. I'm a little bit, it's strange because this is the first proper in-person conference I've been to for a while, so I'm used to presenting at least in pyjamas on my bottom half, so we'll see how it goes in proper human clothes. So I'm from Newcastle University, and what you've been listening to previously, I guess I'm kind of way further down the journey. So what I'm interested in is what triggers help seeking. At what point does somebody go, actually this symptom is a bit unusual, or this seems like something I should get help for? And why do they then seek help? And who do they talk to? And why do they talk to that person? Because what we do know is that there's lots of new treatments, there's lots of new medication, there's lots of amazing things we can do for head and neck cancer. But if the patient with the head and neck cancer doesn't get into the system, then it doesn't matter what treatment you have. You can have everything in the world. You need the patient and the person with the cancer to be in the system at a point where those treatments and medications can actually make a difference to their quality of life. So, I'm interested in head and neck cancer, and I apologise for this first slide because you all know head and neck cancer. I don't need to tell you all about that. But it is a cancer that is increasing over time, and part of that is due to the HPV. But one of the reasons why we're interested in this journey is because the symptoms for triggering help are very vague for a lot of people. They're symptoms that you wouldn't normally seek help for. You've got a mouth ulcer that's not healed. It's not something you might worry about. A lump in your neck, we're used to lumpy necks. We get them when you get a cough or a cold, your neck flares up. These are not things that normally trigger people to seek help. So this is why we're thinking, can we have a look at this journey and see if there's any problems along the route? So the first step is looking at something called routine data. Now every time someone is diagnosed with a cancer, the NHS records the details, and this is called routine data, and they take lots of information and record it in a massive file. It's all anonymised, but we can request some of this data. So I requested all the head and neck cancer patient data to have a look, to just see what pathways patients were coming through, what routes were they taking. And just to kind of give you an idea of the differences, because there are differences in the routes people go along. I've just compared the 2-week wait, which is the kind of urgent cancer referral, with an emergency referral. And what we found in the data was quite interesting. So if you're a white man, and you're aged about 55 to 64 years old, then you're pretty likely to be referred through a 2-week wait, because you match the kind of standard symptoms, the standard patient that a GP would likely see. And go, oh, actually, yeah, this triggers me thinking this could be something more serious. If you're a woman, or if you're not white, And if you're living in an urban area, then you're less likely to come through that 2-week wait, but you are more likely to come through an emergency route. Now, those that come through an emergency route come through because the symptoms have reached a point where they need urgent help. So those patients are often people that have quite a high stage of disease. So we really don't want people coming through that emergency route. We want people coming through the 2-week wait route. We can look at all of this route, and my supervisor will tell you I tend to cry when it comes to numbers and statistics. So this bit was kind of forced on me, but what I'm really interested in is why this happens. And the only way you're going to find out more of these details is by talking to people. So at my heart, I'm what's called a qualitative researcher. And that means I go out and I interview people and I listen to all the stories and all the processes that people have been through. So I interviewed lots of patients. I also interviewed GPs, surgeons and dentists, but today I'm just going to talk about the patient results that I have, about what it was about their route to diagnosis. What were the triggers? What were the things that helped them? What are the things that massively tripped you up along this journey? Because if we can find out where these barriers are, we can put things in place to maybe allow people to get through the system far quicker and far easier. I was also interested in that first point of the diagnosis as well. So I spoke to people about what it was like being told you had cancer, because I'm very interested in the way things are communicated to people. Are they communicated well? Are they communicated maybe not so well? Might need a little help on those bits. Do people know you've got cancer when you've diagnosed? Do you actually understand what it is you have and what you're going to go through? So, I'm going to tell you 2 stories today. I will say the stories have been altered because I don't want any of my patients or any of my participants to be clearly identified. So, the nature of the stories is right, but some details will be changed. But I want to tell you 2 stories, and I want to tell you 2 stories of 2 different pathways, 2 different routes to diagnosis, so you can see how the impact was. You may recognise some elements of the stories in your own journeys. Some of these journeys may be so completely different to your own, but I guess that's the thing, is there are lots of different journeys that happen. So, my first person is Rob. Rob was 45, lived with his wife, 2 daughters. One had just started medical school. He was really fit, he'd go to the gym, he was training for a marathon. He was someone that said he was never sick, not a day in his life. The odd sniffle, but not really anything. In February, he noticed a lump on his jaw, but he thought it was a tooth infection. It wasn't really bothering him. He wasn't that keen on dentists, so he wasn't going to do anything about it. He started to kind of think, oh, maybe I should do something, and this was July now. But he booked a holiday. Now, those of you that have booked a holiday know the last thing you want to do before you go on holiday is go to the dentist. There's a likelihood they're going to pull something out and you're going to be in pain or look a little bit odd in your holiday photos. So he's like, it'll be fine, I'll leave it till I get back. So he left it till he got home. He reluctantly went to the dentist with a little bit of pressure from his wife when he got back. And he was then referred to secondary care. And then 2 weeks later, he got his results after he'd gone into secondary care, and he was diagnosed with cancer. This whole journey was perhaps a reasonably simple journey, but you can see there were massive delays. So from the first point of him having symptoms to the first point, to the actual point of his diagnosis of cancer, was 38 weeks. So now I'd like to introduce Maggie. Maggie's 78. She's got lots of family living by, 3 very active grandchildren. A relative had had a diagnosis of head and neck cancer a few years previously, so she knew the symptoms, she knew the process, she'd been through it. She found a swelling on her neck in December. She instantly thought, this is cancer. This is not right, I'm not happy, something's going on. She went to the GP straight away. Symptoms started to get worse, and she was starting to get an earache, and she kept going back to the GP. She was diagnosed with impacted earwax. She went back to see a nurse. The nurse tried to drain her the wax from her ears, but said, oh, I can't do it, like, this is tricky. I think we need to refer you to the hospital. She then got another referral to the hospital. She was given an appointment at the hospital for 3 months' time. Before this date, the pain got so bad that she went to a walk-in centre where she saw a GP. The GP at the walk-in centre didn't ever say the word cancer to her, but she was convinced that he knew it was cancer. She knew it was cancer, he knew it was cancer, but neither of them were going to say the word cancer. And they were just going to maybe see what was going to happen next. She went back to her GP, which was about the 5th time for this point, and begged to be referred to the hospital. Before this appointment, she attended a hospital appointment with her husband and saw the surgeon. And she said to the surgeon, "Will you just have a little look, please? Because I'm very anxious about this." She did, and then the next thing, the process started and she got the referral into the system. Now, her actual route to diagnosis was far shorter than Rob's. This was 23 weeks to the point of diagnosis. But the stress and the anxiety of this journey impacted her trust with the system. It knocked a lot of her ability to trust the clinician she was speaking to. She, now in survivorship stage, would not go back to her GP because she was really worried about whether she could trust the GP to actually find any recurrence or any symptoms from there. So this impact actually will have a long-lasting impact on her life for afterwards. So what I do is I gather stories. I gather stories and I look at them and I analyse them. I spend a long time looking at them and I look for patterns in the data, and we call them themes. I look for themes that go through the data. So while everybody's journey isn't the same, what you can pick up is themes coming through their journeys. And these themes can help us understand what these barriers and what these facilitators, are there anything there that helps people's journeys? And this is what I come up with. So I like drawing circles. But I'll talk through what these themes are, but essentially, knowledge, you can see, is this big circle in the background, and everything I talk about is based in knowledge. If you've not got knowledge, this is going to be a really tricky journey for you. So knowledge talks about knowledge of cancer. Cancer in general, what do we know about cancer? Then more specifically, what do we know about head and neck cancer? Because head and neck cancer is quite different in the way it presents. Now, if you say to a lot of women, if you found a lump on your breast, there's most women that would go, that's straight to the GP. That is something highly suspicious. But a lump in the neck, maybe slightly less suspicious. My teeth don't fit properly. I've got my false teeth are just a bit oddly fitting in my mouth. You know, it's age, things change. It's not something to really worry about. So it's this knowledge that you don't, if you don't have the knowledge, it makes it tricky. And also knowledge of the healthcare system. For those people that work in the healthcare system, it seems like it makes a little bit more sense. But for those that are not in the health system, my God, there are a lot of letters and words, and it's very confusing. You don't always know, can I go to the GP for this? This is in my mouth. Should I go to the dentist? Should I go to the GP? Who's the best person? Is this urgent? Is this something I should ring my GP about and go on the day? And all of these questions and queries, because if you don't know the healthcare system or you don't have the knowledge to navigate the healthcare system, then that will cause delays for you getting help. And being able to know that, even as somebody in my job, when I've been ill, I've sometimes had these debates with myself. So this isn't something that people outside of the healthcare system, inside the healthcare system also don't have problems with. But understanding how to access things was a big issue. And then symptoms. So we've spoke about the knowledge of symptoms, and one of the big things that came through, one of the major things that came through most of my interviews was, I thought there would have been pain. For a lot of people, the lack of pain was a lack of incentive to seek help. The second pain arrived in any of their symptoms, they were straight to the GP or the dentist. But so many of these symptoms were painless, so there was no reason. Why would you go to the doctor for a very painless little lump? Why? You're told not to waste NHS services. We're told not to waste the GP's time. So you're not going to go for something that isn't actually causing you any problems. And then there's also how other people assess your symptoms. We often might sit and chat down with your neighbour or your friend, oh, I've got this weird thing. Oh, my friend had that lump, but it just turned out to be this, and it was okay, so don't worry about it. Or, in some cases, my friend had something like that, it turned out to be serious, you should go to the doctor. Those people with this knowledge in the community were able to really help speed up diagnosis. And that's, if you had someone in your community that had been through something, it was a lot easier for people to then be given permission to go and seek help. Then we go on to negotiation and navigation. So I spoke about it a little bit with the knowledge of the healthcare system. And part of this is about whether we have trust and confidence in the system we're going through. Do we trust our GP? If we do, we'll go back in 2 weeks when they've told us to take the antibiotics and come back in 2 weeks. If we don't, then we'll be, well, you know, we probably didn't really know. They give you antibiotics for everything these days, so I'll just stay, it'll be fine. We need that trust in our doctors. If we've lost the trust, we're not going to listen to what they tell us. We're not going to follow advice. But we also need the doctors to trust us back as patients. We need the doctors to trust that when we say something about me doesn't feel right, that they trust and listen to that. Because when you have vague symptoms, if you can't say to somebody, I just know, I know in my heart something is not right. And many of the patients said they knew, they had a feeling something wasn't right, but they didn't feel the GP trusted those symptoms with them. And it also goes into confidence. And trust and confidence do go quite closely together, but there is this confidence. Are you confident that what the person you've gone to for help knows what they're doing? A lot of people visiting dentists, we're quite funny with dentists in this country because they ask for payment. A GP doesn't ask for payment. Something about money crossing that barrier that really builds up a little lack of confidence and trust. It's like, are they just saying this because they're going to get a little bit extra cash if they refer me? We're not very good at trusting dentists because that payment system goes against what we're used to. We're used to free healthcare, and as soon as a dentist you have to pay for something, you're wondering whether they're actually extending you for a good reason or just for a bit of extra cash in their pocket. And this is not to say that there's a fault with us feeling like this. This is the way the systems and everything are kind of encouraging the way we feel around here. Fine, next one we've got the C word. Now for many of you, the C word is a word that can be spoken quite openly. We often say the word cancer in conference like this. There's not many dentists that like to say the word cancer. There's a lot of concern that if we say the word cancer, we're going to open a whole can of worms and people are going to have lots of questions and lots of worry. Whereas a lot of the patients I spoke to said if someone had just said to them at that point, I think this might be a potential cancer and we would like to rule out cancer, or we would like to send you for an investigation of cancer, then that might have made them go, right, someone's taking this seriously. But the avoidance of the word cancer and the fear surrounding cancer was quite difficult because people just wanted to be told that they were being taken seriously, their symptoms were being taken seriously. And it's important that the word cancer is used at the point of diagnosis. 2 of the patients in my group that were interviewed did not know that they had cancer. They said they didn't know they had cancer until they turned up for their chemotherapy appointment. And they walked in the room and they went, you only get chemotherapy if you've got cancer. All right, I think I've got cancer. And they told me that they heard lots of Latin words, they heard lots of Greek words, they heard lots of complicated words. They didn't hear the word cancer. Now it may be that the doctor said the word cancer, but a lot of the time, medics are used to using far more complicated language than we are used to hearing. And sometimes you just need someone to say, you have cancer. And that's what a lot of the patients said. They would like a clear, easy diagnosis. There was also the context. We work in a lot of multidisciplinary teams, and for some people this was quite overwhelming. To be brought into a room, to be diagnosed with cancer, and in the room there was a dentist, there was a doctor, there was the trainee, there was a speech and language therapist, there was the dietitian. One lady I spoke to said she felt like she was in a theatre show with light shining on her, and she didn't know that she reacted correctly. She said, I felt afterwards I should have cried. Did they judge me for not crying? They were all watching me. Should I have cried when they told me I had cancer? And there was this pressure to perform in the correct way of a patient. This is not something you need at the point of being told you've got cancer. Am I performing correctly? So there's really, around this point of diagnosis, there's a lot of communication issues that we have and a lot of clarity that needs to be in place so that people are fully aware of what they're diagnosed with. One of the important reasons, and one of the people I spoke to said they didn't seek any help. They didn't get in touch with Macmillan. They didn't get in touch with any specific cancer charities for a long time because they didn't think they had cancer. So they thought they would be a fraud by getting help from those places. They didn't tell their friends and family about their diagnosis, because what if they got it slightly wrong and then their family thought they'd made up the fact they had a cancer diagnosis? So there was a big period of time where people weren't seeking help and support from those around them when they could have done, just because of this lack of clarity around the diagnosis. Control. Control is something very important. There's a part where people often spoke about taking control at the primary care level. They felt like they had to take a lot of control. They had to kind of follow up the GP. They had to take control of their treatment. They had to make sure they were accessing everything they needed. But often, once that referral to secondary care had hit, and they hit secondary care, they were like, take it, this is it. I just want to sit down and I want you to tell me what to do. For many patients, they wanted to just relax at that point. They said, we kept being told to make decisions. The cleverest people in the room were asking us to make a decision about our healthcare. It's like, I don't know, you're the one that's been through medical school, you tell me. And for some people, they very much wanted to hand over that control to someone. It wasn't everyone, but for some it was very important. But more control was taken at the earlier stages in the primary care. Perhaps might lead to maybe issues with around our trust of GPs and primary care services. So the 2-week wilderness is this link between the point where you've had your biopsy and you're waiting to be told you've got cancer. And that was often described as the worst point. The elephant was in the room and it was like, I know I've got cancer, you know I've got cancer, but nobody's going to say it until these biopsy results come back. And then finally, the last one is this emotional labour. And this is the idea that a lot of the burden of emotional labour was taken by the patients. And often people felt like they had to protect their friends and family, and they took on a lot of the emotional burden. They were more concerned about the impact that their diagnosis would have on other members of their friends and family. And in some cases, they were very concerned about the surgeons having to do this day in and day out and how mentally straining that must be for them. And there was a lot of concern on the surgeon. So it's just about this importance of really recognising that there's a lot of patients are taking on a lot of emotional burden at that point. So this is what I've gone through, this is how it's ended up. So I'm never going to cure cancer. I'm never going to make their lives better with curing cancer. I'm not going to come up with a fancy treatment as much as I long to wear a white coat and work in a lab. But what I hope to do with this research is use it as a basis, and that's what we're starting to work on with our health literacy project, to work and see how we can enable patients to get help quicker and how we can enable the healthcare system to come to patients and draw patients into them. Because at its heart, I think it is more, the burden lies with the system more than it lies with the patients. But thank you very much for listening, and if there's any questions later, I'll be happy to answer them.
Thanks very much, Jennifer. My name is Kirsty Taylor. I'm a medical oncologist in the Northern Ireland Cancer Centre in Belfast, and I also have a clinical fellow role through Queen's University Belfast. And we're just going to take a slightly different tack now and change to talk about recurrent or metastatic head and neck cancer, and maybe building a little bit on what Dr. Badger and Dr. Wood have said earlier. So, how I came to treat head and neck cancer. So historically in the UK, it's mostly clinical oncologists that treat head and neck cancer. They give the chemotherapy part along with the radiation treatment. And as medical oncologists, we have a very varied degree across the UK of whether we get any training in head and neck or how that works. So I am Northern Ireland born and bred. We all like to stay there, unless we come to Newcastle, or somewhere in Scotland, but we don't travel very much further. So I went to Queen's University Belfast, and then I did all my training in the Northern Ireland Cancer Centre there. But then I was fortunate actually to get a clinical fellow role that was a joint programme, and this is something new and something that's being developed between ourselves in North America, between Queen's and the University of Toronto. So I actually went to Princess Margaret Cancer Centre for 2 years as part of a fellowship where I worked in the drug development team and also within the head and neck team. And that's really where I kind of got my experience, got my expertise, and which is a nice thing then to be able to bring that experience back and what I learned there to try and help the patients and develop clinical trials and treatments within Northern Ireland. So I'm just going to touch a little bit on ctDNA today, talk about what and why, what the liquid biopsies, and then talk about them a little bit, mostly from my work in potential roles, sort of palliative setting, recurrent or metastatic disease, but also touch very briefly then on where they can come in minimal residual disease and early detection of cancer, because obviously that's where we all want to be. We want to get things at the curative stage, get rid of them and treat them at that point. So what is ctDNA? Well, tumours are made up of cells. The centre of all those cells has DNA, and as the cells go through their various different processes, and cancer cells go through those processes, and different forms of dying fragments of that ctDNA then come out into the bloodstream. And this is known as circulating tumour DNA or ctDNA. So that's really what we're trying to pick up when we do some of these tests. And a liquid biopsy is really what it says on the tin. We're taking some liquid, we're taking some blood from the body, and then we're trying to look in that to see what the components are. Can we pick up that ctDNA that's there? What else is there? What does that tell us about the patient? And I'm going to focus solely on blood tests, but there's advancements in any bodily fluid really. People can use urine if they take some fluid of lungs, pleural fluid, things like that. And lots of different ways for that to be implemented. Screening, diagnostics, staging, what treatment that we're thinking about using for people, and Dr. Badger talked a little bit about that earlier, about molecular profiling and the different wording and things, and how that is maybe what we call tumour agnostic. So it's not definitely about a particular type of cancer you have, but is there a mutation there that we can target with treatment, and then monitoring if people are on treatment, which is what we're going to talk about today. So why liquid biopsies? Why take this blood rather than just going in and doing a normal biopsy, or a standard biopsy maybe is what we should say. So a lot of people in this room will have had biopsies, they're not fun, they're invasive, head and neck cancers come with difficulty with nodes, different areas of tissue, not necessarily easily accessed. If this is a recurrent metastatic setting, people have maybe had surgery or radiation before, which makes that tissue a little bit more difficult to get at. It's very localised, we're only looking at that particular area in the head and neck. We're not maybe looking at other cells around the body, lungs, liver for example. Yes, it all comes from that squamous cell cancer, but it changes over time and develops new mutations, and we're not getting access to those with a localised sample. It's time intensive, it takes time. We have to organise surgeons, theatres, if it's a CT guided biopsy, interventional radiologists, all the different space, and there's a lead in and a wait time, and the 2-week wilderness as people are waiting on results and things to come back, as well as getting the biopsy scheduled. And then these things do come with a little bit of risk as well, pain, bleeding, different things like that. So liquid biopsy, on the other hand, is something that we're all aspiring to in different areas. So it's minimally invasive, these things are easily obtained, it's a blood test, same as what people would have routinely before chemotherapy, etc. It's We hope that these things will become more comprehensive, so that sort of heterogeneity as tumours change to different parts, develop new mutations, that we'll be picking those up. It's quick, it's easy to do, there's still a bit of a wait to get results obviously, but it can be done there and then in the clinic, and there's reasonably minimal risk. It's just a blood test which people will probably be getting anyway, and we can add this on. So for head and neck disease in the recurrent metastatic setting or any settings, it's also obviously a multi-modality approach, and sometimes the more of those we add together, the better chance of cure we have for patients. So surgery, radiation, systemic therapy, and as a medical oncologist, it's the systemic therapy that obviously gets me very excited, and particularly the immunotherapy. And Dr. Wood talked about pembrolizumab or Keytruda earlier, which is really kind of our standard first line if we can give it to patients. Rather than chemotherapy for a number of reasons, and I'll explore that a little bit later. But I'm also very grateful to Dr. Wood for explaining how this all works, because the immune system and immunology is very exciting, but it's very complicated. So I'm glad she explained it all to you earlier, but in my mind I always think about it really simply. I just think that these cancer cells are really clever. They found a way to evade your immune system, to hide from the immune system, to allow them to develop. And the cancer cells, in certain mechanisms, have pressed that stop button on the immune T cell to stop that attack. So the drugs that we think about, the checkpoint inhibitors, so Keytruda, pembrolizumab, which is now the first line setting, and also nivolumab in the second line setting, which is kind of more what I'm focusing on today, really inhibits that stop signal, takes the brakes off the immune system, allows that T cell to be reactivated, and then really use your own immune system to recognise those cancer cells as foreign. And so Keytruda is now in the first line setting. Nivolumab was licensed first in the second line setting, and some of this work is from when I was in Canada, so that's what was authorised by Health Canada at the time. And it's still what we use in the second line, potentially, if somebody has had chemotherapy within 6 months and they're no longer sensitive to that, or what we deem platinum refractory. And when this first came out a number of years ago, it was really exciting, because it was the first time in recurrent or metastatic head and neck cancer that we had a treatment that was going to improve overall survival for those patients that had progressed after their chemo. And as medical oncologists, we get very excited about these curves, Kaplan-Meier curves or survival curves. And what we really want to see is when there is a split in those curves, we see that there's an advantage. But the big thing with immunotherapy, and what is a lot of focus and what we're very excited about, is this little bit at the end where it tails off, the blue line, where we see that that continues. So that is showing us that a number of patients are getting what we call a durable benefit from chemo. So that is extending beyond that initial response. And those patients, that can continue for long periods of time. But the problem is, at the minute, we just don't have a good way of picking out who those people are. And as we said, about 1 in 5 people benefit. If I have 5 patients in my clinic in the morning, I can't pick out which one patient is going to benefit from that treatment and what other 4 are not. And that's something that we're really looking into and trying to explore, as well as getting the newer treatments and adding on additional treatments to see if something added to that, does that increase that response rate and make things better. And the other really nice thing about these immunotherapy drugs, and what can be seen from this CheckMate 141 study with nivolumab, is the lighter green colour is the nivolumab. So this was really the first kind of study that truly told us that patients had a better quality of life on these immunotherapy studies rather than with second line chemotherapy drugs. So the quality of life got better rather than worse for the patients that were in immunotherapy, and also we saw that the time to deterioration or feeling less well was much longer when they were in immunotherapy rather than chemotherapy. So at the time this was very exciting, and we obviously see similar things with the Keytruda and pembrolizumab just in the first line setting, which is obviously even better again. So at this point in time, we assess treatment for any patients, whether it's chemotherapy, immunotherapy, targeted therapy, whatever we might be using, by CT scans. And as we said, we've no good way of picking out which patient this is going to help and which patient this isn't. So these are CT scans up on the screen, and if they are really looking from your feet up. So if you were lying flat, this is looking from your feet up. So right hand side, left hand side, and the black areas are the lungs. So you can see patient A on the top, patient B on the bottom. These patients were essentially the same. One was a little bit older than the other, but not by much. They both had oropharyngeal cancers that had recurred with lung metastasis. They both were P16 positive. They both were PD-L1 positive, which is one of the markers that we look for these immunotherapy drugs. But we can't say before they start who's going to benefit and who's not. And very occasionally with immunotherapy, we actually get some interesting responses where people can actually have disease get slightly bigger at their first scan, and then it responds later on. And again, we've no way of differentiating out who that patient is versus whose disease is going to get bigger and continue to get bigger. Different if it's new disease that has appeared, but how can we tease out these patients, or how do we know? So we had what we call a correlative study, so this is non-interventional, so we weren't changing treatment based on this, but masked or multi-omic assessment of squamous cell cancers receiving systemic therapy. So these were patients with recurrent or metastatic cancer who were fit enough to get treatment, obviously. Platinum chemotherapy first line was the standard of care at that time, and then immunotherapy with nivolumab in the second line, or as part of a clinical trial within our drug development team. And obviously, what we were doing then was looking at their liquid biopsies at various different times on treatment to see, could that tell us a little bit more about what was happening in their cancer at the various different times. We got a baseline sample, an early sample after their first treatment, so after 3 or 4 weeks, because as we monitor things with CT scans, that tends to be after 3 or 4 cycles, 3 months. It's a bit of a time to wait, we don't know early on. So a sample at start, after an early time point, at the same time as their CT scan, and then if and when their disease changed. And we're working through these at the minute and really focusing on the early time point. And again, as Dr. Badger mentioned earlier, there's lots of different ways of looking at DNA, lots of different language around this, molecular profiling, panels, next generation sequencing, etc. And we use 2 different panels in this analysis. One is kind of what we call a fixed or an off-the-shelf panel that can be just applied to those samples straight away, and that's this CAP-seq, so Cancer Personalised Profiling by Deep Sequencing. And this is actually squamous cell optimized, albeit we can take it off the shelf and use it straight away. And this was developed by looking at some of the huge cancer databases that we have, the TCGA, and picking out the mutations that we see most commonly in head and neck cancers, and making a panel to make sure it had all those included, covered them all, that we weren't going to miss something. The panel was developed, and then we can apply that to each of our liquid biopsy samples as the patient moves through their treatment to pick out the mutations that are there for each individual patient. The other panel we used is very exciting. This is personalised and bespoke to each individual patient. So this is made by a company called Innovata, and it's actually for residual disease and recurrence. So it was developed in the minimal residual disease or curative type setting, but we were obviously using it in a slightly different scenario. And this is individualised for each patient. So what they do is they go back and take the initial biopsy sample from the initial surgery or biopsy, and they have whole exome sequencing. So that is looking at the parts of your genome that code for proteins, to see what mutations there are individual for that patient and their tumour. And then we followed those specifically at each of the time points that we had. So there's lots of exciting technology coming through in this area. And the patients that we had in the study were representative of sort of recurrent and metastatic head and neck population. Some people were smokers in the past, lots of different areas within the head and neck covered there. Some patients were HPV positive, some weren't. Different numbers of metastatic sites. And then about a third of our patients had the kind of standard first-line chemotherapy, and then about 2/3 had immunotherapy in the second line. We had a small study with 53 patients, and 49 of those we were able to detect the ctDNA and take them forward. So the questions that we were really asking ourselves were, is there something earlier than those CT scans that we can tell? And we wanted to know if the amount of ctDNA that a patient had at the time of the start of their treatment indicated in any way what was going to happen. And these curves, again, that we love as medical oncologists, but again, I talked about the sort of nice separation, and that indicated that something was going to help. And with all of these, we don't actually really see much separation. The amount of ctDNA there, just at the baseline, didn't really tell us very much. So does that change? If that changes when somebody's on treatment at that early time point after 3 or 4 weeks, does that give us a better indication of what's going to happen? And actually, very interestingly, we see that it did. So the blue lines on the top of each of these show the patients that had a decrease in the amount of ctDNA that they had between those first 2 time points. So, you know, this is sort of an experimental study. We're looking at people in standard of care treatment. This wasn't going to make us alter their treatment in any way. We're just trying to watch what happened and see. But this is something that, you know, gives us a bit of food for thought, and is there something else we can do there to better identify those patients? And at the minute we're using their CT scans to tell, so can that be worked in some way? So this just represents that change in a slightly different way. So this graph you can see on the side, all those bars along are each individual patient. That's their time on treatment. Each individual patient is listed there, and the little sort of line graph in between, the red are the patients that their ctDNA increased or got worse, and the blue are those that decreased. So you can see in general on the bar graph on the top half, those patients tended to do better when their ctDNA went down. So as I mentioned then, obviously we're looking at CT scans at the minute. So can we put these 2 things together? Is there information there that will better tell us how to pick out these patients, how to know who to continue with treatment, what's the right thing to do for the patient? And we did see, again, the separation in these curves. The nice dark blue line at the top are the people that we saw things getting smaller on their CT scan, and their ctDNA going down obviously did much better, and that's kind of a no-brainer. Those were the 2 things that showed benefit. We didn't really have enough patients that in the study that responded, but their ctDNA got better to draw any definite conclusions. But we did also see that those patients that their CT got slightly worse, if their ctDNA dropped down, they actually had a slightly longer overall survival in terms of time. It wasn't significant when we looked at it, but they did benefit in some way. So hopefully, you know, we give palliative treatment to help control disease and disease-related symptoms, to keep people as well as we can for as long as we can. So is that an indicator that they will be feeling a little bit better and have a little bit of a better quality of life? And this has been shown across some other studies. This is actually another study that was done through the Princess Margaret with Keytruda, looking at some of these patients. And again, this included, there's 100 patients in total. There was one cohort, 20 patients were head and neck, and there was a multitude of other cancers in there as well. But again, they saw that this combination of a reduction in the amount of tumour DNA that was there, along with the reduction in the CT scan, people did better. So this is all very early stage, but it's something that, you know, gives us some hope that there might be something there to look at more closely. And this is slightly unrelated. This is not to do with tumour DNA at all, but we just wanted to know, was there something else there, some information that we have readily available that we can help identify patients a little bit easier if you're going to benefit or not? And these graphs are looking at what we call a neutrophil lymphocyte ratio, so white blood cells in the blood, and a platelet lymphocyte ratio. So platelets and white blood cells are checked every single time before patients have treatment, so you're not having any more blood tests. It's something that's readily available to us in the clinic, and these are kind of considered, when they're divided into the ratio, as markers of inflammation, circulating inflammation. And actually we saw that when people had a neutrophil lymphocyte ratio or a platelet lymphocyte ratio below the average or the median level, the middle level of everybody that was included, that they did a little bit better. So these are all just things that are experimental. We would never change our practice on it, but looking at things that are easily there, the patient doesn't need to undergo any extra tests. It's something we have available. It's very cheap to do. Can we in some way use this or build it into the other tests that we have to give us some more information? So just to mention sort of ctDNA in the UK and recurrent metastatic head and neck cancer, and where that is, the main area that we, certainly in Northern Ireland, have access or availability to this at the minute is the Target National Study, which is run through the Christie Hospital, and that patients who have progressed beyond their standard lines of treatment or don't have other options left can go and have either ctDNA from blood tests, or we can actually send their tumour tissue as well to get this next generation sequencing or to look for mutations. And then if we pick up something that we think might be targeted by another type of drug, that's maybe like trastuzumab for HER2 mutations in breast cancer that we talked about earlier, or can we use that for these patients to help gain some benefit? And then this study, after they have their liquid biopsy, actually matches people to trials across the UK. And just the very last couple of slides, just to mention then, recurrent metastatic disease is obviously my area of interest, but minimal residual disease and detection of recurrence is really where we hope to apply a lot of this ctDNA monitoring to see, can we pick things up sooner for patients that are treated in the curative setting, such that if there is going to be a relapse, we can pick it up sooner and treat that. And these are 3 different patients that were sequenced with RADAR, which is the personalised bespoke panel, so taking their own tumour and looking at the different mutations within that. And probably the most interesting of them all actually is the top patient, patient A there. So they actually had 2 synchronous tumours, 2 tumours at the same time when they were diagnosed, an oropharyngeal tumour and a hypopharyngeal tumour. They underwent surgery, and we see that the hypopharyngeal tumour, from the ctDNA, the mutations that were present in that were completely cleared at the time of surgery. They went on to have adjuvant therapy anyway. We obviously we're looking at the ctDNA in hindsight, it's not altering our treatment at this point. But there was persistence of the ctDNA in the oropharyngeal tumour. They had their adjuvant treatment and it continued to rise. There was nothing to see on CT scans or scopes at that point. So you can see that there's about 8 months there where that ctDNA is positive before we really see it developing on the CT scan. So that's another place where these ctDNA assays are really going to come into their own and really help our treatment planning and decision making, and hopefully improve cure rates for head and neck cancer. And this, just the last one, is exactly the same kind of scenario, where again patients who had surgery then had their ctDNA monitored afterwards, and again probably the bottom curve is again the most interesting. We like to see things splitting for us as medical oncologists, and the blue line there is patients that partially or completely cleared their ctDNA, had much better overall cure, whereas those that did not clear their ctDNA, you can see how quickly they dropped. So these are just areas that are really exciting where this is starting to be developed, and hopefully we'll be able to benefit patients in their treatment in the future. So I just want to say thank you, and particularly my main thanks is to the patients and their families that got involved in our studies and allowed us to take those extra blood samples and to give us a bit of an indication what's happening here and hopefully advance things. And this is the head and neck team in Princess Margaret in Toronto.
Very different approaches, but both equally important. Before I open the floor, I have 1 or 2 questions. You made a very interesting comment about the dentist not mentioning the cancer word and being very particular about not doing that. And I don't know whether it comes from the fact that there is quite a sort of fear or culture of sort of litigation, and people don't want to say things that they're not sure whether there will be cancer or not. What I've seen recently as well in terms of training the undergraduates, people who are becoming dentists, is that the emphasis is changing. It used to be tell the patient they might have cancer and send them to the hospital, and now they're being taught and trained, no, don't come in, do not say they got cancer because they may not have cancer, and you cause that additional stress. So I don't know whether it's because there's been a change of culture or how they're being trained, etc. I'm not sure, that's just my opinion. I don't know what you think.
No, I think there's definitely a lot of fear around mentioning the word cancer. I think we're quite cautious about, especially clinicians are very cautious about using the word because they don't want to cause any undue stress. But actually a lot of the research that's coming out with patients says they want the word cancer used. They want people to be very clear about what treatment they're doing or what they're investigating, what they're looking at. And I think this is just kind of one of those situations where we're not linking the research with patients and service users and the research around anxiety around the clinicians. And I think actually if we mix it together, we'll realise that we could train clinicians from a very early stage to cope with the anxiety of using the word and the anxiety of giving a diagnosis or a potential diagnosis when they don't know the answer. But I think that it's definitely a training thing, but there is a lot, as litigation becomes bigger, there is a lot of fear around that. But definitely from the patient side, I'd say most people are normally saying, please use the word. It makes me feel safer to know that someone is looking for these things. And, yeah.
And you mentioned the differences in terms of like middle-aged males being seen, more likely to seen within the 2-week period, and I wondered if you looked at or see any differences in terms of people who were not native English speakers and whether that made a difference, and was it similar or worse than sort of females and ethnic minorities, etc.
When we look at the data that we get from the NHS data I was looking at, routine data sadly has been incredibly poor with taking on a lot of ethnicity details, which is why when I was saying I said kind of non-whites to white, because actually the data currently as I wasn't able to look at specific ethnic groups. But there is a small amount of research that showed that certainly within London, there's a lot of, some groups of Asians will be more likely to go to a dentist. And actually those that go to a dentist, I didn't show that there, but if your cancer's caught at a dentist, it's more likely to be caught at a very early stage. So it's more likely to be stage 1 cancer, in which case the treatment's far less difficult from there. So we still don't know, unfortunately. Previously, the data the NHS has collected has been very poor around ethnicity. But actually women and ethnic groups appear to use dentists far more frequently and often than men. Sadly, men are very poor healthcare users to their detriment.
The cost thing was quite interesting because you said people will have to pay at the dentist and maybe go to the GP, that's more likely, but dentists, like you said, are much better at spotting it because they've been trained and they see it a lot more, so they're much more likely to pick it up at an earlier stage than GPs.
Yeah, definitely.
And you mentioned, you were talking about immunotherapy and how we give these medications. I was thinking in terms of the CPS score, does it matter whether it's greater than 1 or greater than 20, and do you see that response in treatment as well based on CPS score, or pathologists just wasting their time doing all that scoring for you guys?
Yeah,
so the most straightforward thing to do.
Yeah, so I suppose with our license, the NICE license for Keytruda in the first line at the minute is if the PD-L1 score is more than 1%. In Northern Ireland, we just have pembrolizumab alone. We don't have the combination with chemo, which I think Scotland have. So there's still a bit of discrepancy there. But I mean, I think there maybe is some discrepancy that we see with the PD-L1. Certainly the trials, when you divide out the evidence, there is slightly better responses when people are more than 20%, but it's not huge differences. And with nivolumab, we don't have to have that PD-L1 back. So even if somebody is PD-L1 negative, we can still use nivolumab in that scenario. And we do still see that people respond. So it's a bit of a guide, but we would like to have something better.
Thank you. Anyone's got any questions? There's one question.
One here.
Thank you so much for interactive lectures for both sides. I would like to ask something more about liquid biopsy. Now, is this something getting used quite so often? And is it something, if it's getting used, can it be used for post-operative patients as well once they have surgery to monitor for early recurrence rather than late recurrence for them? Please, thank you.
Yeah, absolutely. So I think at the minute it's not something that's routinely used in practice. It's mostly within the clinical trial space. Some of the areas that we are routinely using in clinical practice are more in lung cancer, with some of the targeted treatments looking for DNA mutations that can be specifically targeted with drugs. In the head and neck space, I think at this point in time it's all within, mostly within the research area, but certainly that early detection and early recurrence, and exactly as you say, the sooner that we could do it after surgery, the better. And everything that I was talking about was non-interventional, so actually we looked at the ctDNA in hindsight with the patient's outcomes, whereas a lot of studies now are looking at things in real time and developing interventional studies. So if they do see a change in that DNA, then scoping patients earlier, CTing them earlier, thinking about whether they need an alternative treatment. So that's exactly where things are going. The earlier we can do things, the better we can treat people, better cure.
Thank you. Please keep it going. Thank you.
Hi. Would you just say that there's a definite inequalities in diagnosis of head and neck cancer?
Yes.
Mainly because we're working on a project now just about to start with the NHS. And I'm just interested in what your findings were about the inequalities, because whenever I go into a waiting room, and I go around a lot of waiting rooms, there's a lot of white British faces, but you very rarely see an Asian person sat there or another inequality sat there. What do you think? Why do you think that reason is?
Well, we definitely know there are inequalities. Some of the research didn't speak about today is I've looked at the inequalities within the pathway. So we know that women and non-white are less likely to be diagnosed as quickly as other people. Don't know why currently at the moment. I think there are some things around, I think there's a lot of research now around the inherent racism within our healthcare systems and whether there are things around there. We know that certain communities are more at risk of head and neck cancer, beetle chewers, which is more of a kind of Asian community, then they're more at risk. There's a lot of research around trust, and whether the GPs or the people that we go to are people that we trust, and are we seeking help from people? Are we making our services available? We don't often have services in other languages. If you don't speak English as a first language, the NHS is an incredibly difficult system to negotiate. We have the same issue with people who have hearing loss, people who are blind. They have difficulties accessing the system, accessing our healthcare system. I couldn't tell you why. I think there is a myriad of different influences on there. I don't think there's one single thing, but I think we do have to accept that we have significant inequalities within our system.
So could it be down to culture or education or a bit of both?
I think potentially a bit of both. I think there are definitely cultural differences in how we access healthcare and who we go to. And I also think that there is an education aspect from the clinicians and the system. I think the emphasis should always be placed on those that have the most power, and that's the healthcare system. So the emphasis should be on the changes being there rather than too much emphasis on kind of trying to change patient behaviour. I think we focus a lot on changing patient behaviour when actually we need to bring the system to meet the patients rather than trying to get the patients to come up to meet the system. And I think that's why we're kind of not quite getting it at the moment.
Perfect, thank you.
I think there's a definite difference in culture as well, Chris, because I have been working with groups in Pakistan where mouth cancer, head and neck cancer is one of the most common cancers. And women are always much less likely to come in sooner. They always come in late stage. Even things like toothpaste and basic things are thought of as like beauty products. And there's also sort of a religious component to it as well, where people feel like that they've got the cancer because they did something wrong in the previous years, and this is their punishment for it. People also believe in God, of course, and think that this hardship is from God, and that it's for me, it's a test for me, and that I have to actually fight this and just put up with it. So there's a lot of different sort of mindsets and things that people are thinking about.
But it's a thing that we need to break down.
Absolutely.
So maybe with the team that we've got at the back here that's working on the budget, we can take this outside here, talk more.
Hello. I don't want to ask a question, I just want to make a comment about the dentists. I was diagnosed during COVID, and when I rang my doctor, I'll give you my spray. I said, no, 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 doctor, and etc., etc., the lump, the whatever. He said, no, 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 I will give you a mice spray. And I said to him, do you think it would be an idea for me to go to my dentist? He said, yeah. So I went to my dentist and luckily enough got an appointment. Now, I had my suspicions and my dentist said to me, she was a female, I am very worried. I am very worried and I am going to fast track you to Derry where I had my treatment. I didn't need to be told. She didn't need to say the cancer word to me, but I was eternally grateful to her. I think you made a comment about people having suspicions about dentists and asking for money or whatever. That never occurred to me. I suppose I have a father who's a dentist, I have a sister who is a dentist who worked in the hospital, so that was never, something that I thought about, so it was a new concept to me, but I take that on board. But certainly I am rooting for the dentists. And I have to be very honest and say my confidence and trust in GPs has gone completely and utterly. Unfortunately.
No, and I don't think that experience that you've had is unusual. And I think when a dentist catches, one of the findings I didn't discuss is when a dentist finds a cancer, what you often find is that person is never leaving that dentist again. That dentist, I've known a dentist retire and he said he had patients cry because they'd gone through a whole system with him, and that trust when they discover something, that is a real trust-building event, but unfortunately, that lack of trust now with your GP, and I think the sad thing is that could potentially have an impact on people's future illnesses and health. And I think that's what we really need to talk about and consider, is the long-term impact.
But also our dentists in the NHS get paid to do an oral screening.
They do.
Every time. And I still find dentists that don't do it. And when I find them, I ring and ask whether they're going to reimburse the money back to the NHS. And they actually find it very upsetting them asking them questions. Because I think what they're doing is taking money and not offering a service. And I do think if anyone ever goes to a dentist, and I've always believed that our dentists have our mouths open more than any other health professional out there, if they don't offer you an oral screening at the end of that service, then you should say to them, I'm entitled to an oral screening. Different with private dentists, obviously, because they are private. But if you're just an NHS dentist, and there's not many out there, unfortunately, now, they are going to be coming like a hen's teeth, but when you do find them, they should be doing it. I had one dentist owner say to me, yeah, but I've got 12 dentists. If you take 60 seconds every patient, and then you multiply that over my dentist, and you multiply that over a year, that's a year's salary. But then you've been paid it. And he actually sent that in an email, so I sent it to the Dentists Association. Because that's not right. So I do think there's a lot of work to be done in our dentist line. The majority of our dentists are very good.
Yeah, and a lot of the dentists I interviewed, I will say, were very... it was slightly terrifying interviewing a dentist sat in a dental chair because that brought out all my anxieties. But I will say they were all incredibly knowledgeable, incredibly helpful, and they are a profession that are open to change, but I think we do perhaps need to push for change, and I think that's why the charity and things are very good for this, because we do need to push for what should be happening. And I'm highly suspicious if my dentist doesn't poke my throat at least once during my...
I mean, my dentist will always say that I'm doing an oral screening for cancer. And what he said to me is that when he tells them, do you know what, everything's good, everyone smiles.
Yeah.
But the odd one he has to say, well, I'm going to refer, if it is cancer, they love him to death. all the way through. So, thank you.
Thank you.
Thanks a lot for your talks and for the interesting discussion. And thank you to all of you for engaging and asking all the questions.
My job just to introduce the final session of the afternoon.
Just do it, Mike, you're not on speaker.
Try again. So, it's my pleasure to introduce our final session this afternoon. So our speaker this afternoon is Aliska Rotner-Glover, I hope I pronounced that correctly. She's come over from Prague and she's going to be speaking to us about her experiences looking after patients going through proton beam therapy. She's an ENT surgeon over there. Again, the same format. We'll listen to Aliska and then there'll be an opportunity to ask any questions. So.
Good afternoon, ladies and gentlemen. First, I'd like to thank you for the opportunity to present here our work. So, as Dr. Nagit said, I'm not a radiation oncologist, I'm otolaryngologist. Can I ask you for my presentation? Yes. And so I will focus more on the otolaryngologist point of view after a short overview, which are slides from my radiation oncology colleagues. So let's start. The institution was built for proton beam irradiation. It's in operation already for 20 years in Prague, and to date, nearly 9,000 patients have been treated. And nowadays it is necessary to cooperate in larger groups, so we became a member of National Oncologies and we closely cooperate with other university hospitals, of course. So in Prague, in the building, there are 4 irradiation rooms. They all work with the pencil beam technology. And in 3 of these rooms, there are special rotational systems called gantries, and one uses a fixed beam technology. The first abbreviation means imaging guided radiotherapy, which means that the target volume is during the treatment session is many times being modified according to the changing volume or to the changing size of the tumor. With the vision RT, we precisely check the position of the patient to prevent any motion changes, and of course in the institution there are the basic imaging methods like MRI, CT and PET-CT scans. And in such institution there must be close cooperation of many specialties, so there are more than 130 employees consisting of radiation oncologists, of course, clinical oncologists, radio diagnostics, nuclear medicine specialists, For children, there are necessary anesthesiologists, urologists, ENT specialists, and so on. And besides physicians, there are, of course, many radiology assistants, and large work depends on nurses, of course. And there is also a large client service, which facilitates the first contact with the patient, and each patient gets an assigned coordinator who helps with completion of documentation, organization, some administrative solutions, and they also help with accommodation assistance near the center. And so we came to the basic questions why protons can be very good for some of the head and neck tumors. So these tumors basically have comprehensive target volumes, but they are very close to fragile structures that need to be spared. And also, head and neck some of the head and neck tumors have high curability rate, so we need to think about late and very late effects. And don't worry, I'm not going to go in details because, by the way, I don't understand it because I'm not a laryngologist. But in general, protons can deliver very high energy precisely to the spot of the tumor. This is caused by a Bragg peak feature, which easily means that protons bring all their energy to the tumor, but the dosage falls steeply in the tumor, and the other tissues are affected very little. There is a very low dose in the surrounding tissues, and they have the same radiobiological effectiveness as photons. This slide shows which locations in head, of course, are necessary to be spared. We need to spare brain tissue to prevent cognitive dysfunction. We precisely try to spare optic nerves to prevent latent neuropathy, and we try to spare cochleas to prevent hearing loss later on. And of course there is a spinal cord, which if it's not spared may lead to myelopathy. We like to spare pharyngeal constrictors to prevent dysphagia later. And we have a very low number of patients dependent on gastroscopic tube, or practically no patients with this. And we practically don't see chronic laryngeal edema in the post-treatment time. Of course, with protons, it's quite easy to spare the other side of the tumor that is being irradiated, so it lowers the amount of acute mucositis. But fortunately, we must say that still there are locations that cannot be spared. The tissue that is very close to the primary tumor is very difficult to spare. This is a question of paranasal sinuses around orbit and these regions. And the situation is also very complicated in midline tumors. So everything has been said already early on, so I just quickly go through and to say that even with proton radiation, patients do suffer from acute toxicity, which is mainly mucositis causing dysphagia, and some people experience mucosal swelling with laryngeal edema. Skin reaction is almost the same in the acute way, but heals quickly and properly. There is a very progressive scar tissue development, and some people having their tumors in the parotid region develop external otitis. And so here is the overview of late toxicity, even after protons. Patients do have xerostomy still, but it goes away like a year after the treatment. They do have post-radiation dysphagia for a few months or years, it depends. We do have patients with cataract after a few years after the treatment. Skin fibrosis in the neck is almost a question of the past, because now we can spare the skin in the neck very nicely. Hypoacusis is what we are trying to accumulate data about hearing before and after, a few years after the treatment, but we don't have enough data to say the results. We have one patient with carotid artery stenosis, and unfortunately some patients do have osteoradionecrosis. And so which cancer sites are very suitable for protons? There is tonsillar cancer in the post-operative way. Paranasal sinuses are very suitable to be radiated by protons. Nasopharyngeal cancer as well, salivary gland cancers, and there are also some benign tumors which are very recurrent or invasive. Recurrent is, for example, pleomorph adenoma, and protons are also very suitable for recurrences in the ENT region. And there are benign tumors, paragangliomas, or chordomas, which are invasive. Well, so here I just present here a few of our patients. This is a young athlete. He was quite young in the time of diagnosis. We are usually proud to present him at conferences. You will see later on he came with an enlarged neck mass. Then he was diagnosed with squamous cell cancer from the needle biopsy, and then the primary was found in his right tonsil. He underwent surgery and then chemoradiation with a mild or average toxicity. Here is his radiation plan, and what is almost unbelievable for me that he underwent European Championship for Ironman less than half a year after the treatment. I couldn't understand. And he's in complete remission. I think this year it was the 5th year after his treatment. Another patient is with nasopharyngeal cancer. He again came with quite big neck mass. He received a standard procedure with chemoradiation, and he was not in a good condition at the end of the treatment, especially after the adjuvant chemo, which is standardly given to nasopharyngeal person. He had very large difficulty swallowing, some infections and things like that. This is his tumor in the picture, but you can see that his dysphagia is gone now. Okay. And the last patient I'm presenting here is a young soldier. He was diagnosed with a quite large tumor, and again received a standard procedure. But I've given him in my presentation because I can show you nice pictures. This are the starting MRI and PET. And here I can show you the endoscopic finding of the tumor at the beginning of the radiation, and the second photo shows what it looks like after the 7 week, at the end of the radiation. And this man is, just to show that re-irradiation is possible, he received first, because of his carcinoma of the floor of orbit, he received first photon treatment. Because of the recurrence, he received re-irradiation by protons, and then, because of another recurrence, he had re-irradiation with protons. So it's possible, unfortunately. This is his MRI scan. And so here it's just to present you some of our papers, because nowadays you can't do without presenting your work. So this was a paper from 2021, and here is a case report, another paper from last year. So in conclusion, I would like to say that proton beam therapy is suitable for some of the head and neck tumors, with excellent results for patient survival. It's typical that it can deliver high doses per fraction with very high precision. The acute toxicity is normal or mild, and the chronic toxicity is maybe lower in some aspects compared to other methods of radiation. Thank you for your attention.
Thank you for the talk. It's fascinating to hear about something that is getting some attention now in the UK. Do we have any questions from the audience?
I'll start with one. If Proton is so good, and I've been to Prague and I've seen your unit and it's phenomenal. I can't believe what goes on behind the scenes to get one little beam to the actual point that it is, but you need to go and see that if you ever get a chance. But why is it not used more widely in the UK?
Because it's much more expensive. And I don't know.
So, can you help, Michael? Why do we not? I mean, I know that there's Proton here in the UK, but it seems that head and neck, just the same as everything else, gets left on the side. Unless you're a child with head and neck, you're not going to get Proton.
Yeah, I mean, I think we've only sent one patient for proton beam therapy, and that they had to go to, this is 7 years ago, they went to Florida for it. It was a young girl. I can't answer that question other than to say that, you know, cost is a significant factor. And I think sometimes the NHS is a bit slow to adopt new ways of doing things as well. There's a bit of inertia. I think the last...
That more than 100 British patients have been already radiated to Prague.
Yeah, that's interesting.
So,
for head and neck cancer or for...
No, not only for head and neck and prostate.
but if it, I'm not being horrible, but it annoys me when I hear that it's down to money. Because when it's someone's life, how can you put money on someone's life? And I know how polys work, I know how that all works, but at the end of the day, we're talking about a person, not a patient. I think sometimes the NHS, not yourself, Mike, but the NHS management team see a patient, and what they do is they put a price on that patient's head until it becomes a member of their family. Then all of a sudden I think they do change their attitude, and I think the NHS, the governing bodies who deal with budgets, should realise a patient is a person, and that person has a family, and that person is a human being. And I do think sometimes in the NHS there's a lot of postcode driven treatment, which is wrong. And I've also seen a lot of Blackpool's got a great team, you know, you've got a great team, but you know, you go into other areas and it's not great. Nottingham do a great thing for After Effects, yet you know, we don't adopt it right across. No one talks in the NHS like I don't think they should do. And Proton has been around for I don't know how long, since I've had treatment. And everyone keeps saying, well, it's, you know, it improves side effects, it improves everything. Yet here we are still with no Proton. And Rutherford have just closed 4 centres that the NHS could take over and start doing Proton. I don't understand it, sorry. I'm not having to go at you, Mike, but I do think that NHS should have a lot of embarrassment at the moment.
So actually talking about this last year, I'm also an otolaryngologist, and we had the proton beam panel last year. But we have the same problem in the United States. There's not a lot of availability of units, and really for people getting upfront radiation, we typically do the photon beam therapy, where essentially it's available at most cancer institutions, and we've usually kept the proton beam therapy in reserve for re-irradiation, as you had mentioned. Certain nasopharyngeal tumors or sphenoid tumors, things that are right in the center of the head where we need to spare the optic nerves, which you really can't do with, you run some risk with proton beam, but you run a huge risk with regular photons. So it's really been used more. There is a financial aspect of it. The centers are expensive, but it's also really come down to availability to some extent as well. So we usually use it for like the re-irradiation, so we can avoid, I mentioned earlier the damage to the normal tissues. With proton beam therapy, you can really limit that if you have to re-irradiate an area. So that's been one of the reasons.
Still down to money, I don't care what anyone says. So any other questions? It's an interesting subject, and I think it's one of those debates and subjects that will go on and on and on and on. Unfortunately, we're going to run out of time, so back to mine.
Actually, I've got one little question. Let's get. Do you have any idea of? You said that some of the late effects might be that of the proton beam. Do you have any sort of numbers or particular?
You mean what to improve?
Do you know if the percentage of patients getting osteoid necrosis, for instance, is lower or?
No, it's not changing the percentage of the patients with osteoradionecrosis, but I think during these 20 years, for example, the dosage for the neck skin was lowered as much as possible, and really, I remember at the beginning, the patients did have a hard skin on their neck, and now it's like you can't see that the patient was irradiated from outside. You don't recognize it on his neck. And
do you think fibrosis is a significant improvement?
yes, and this is the advantage that you can precisely put the energy to, for example, to the lymph nodes and prevent the skin.
Well, I've had a thoroughly enjoyable day today. It's been quite invigorating for me, just listening to a very different type of conference to the one that I would normally go to. So many thanks again to Chris and his team for allowing us to host this year, and we look forward to seeing you all again tomorrow.
So just one more announcement. The people that are going to the speakers' dinner, the bar is open, even though you're not going to be eating until 7:30, but the bar is open. I know you'll enjoy like a bar that's open early. So enjoy the rest of your evening. If you go to speakers' dinner, we'll see you there. Tomorrow, again, if you can be in early doors, we do start at 9, so we can get away early tomorrow afternoon. But we've got a packed day tomorrow again. Please turn up, please come and support the speakers, support our sponsors, and I hope you've enjoyed the first day. Thank you very much.